So the next question we want to ask, because you want to do something useful, you discover it based on artificial system. You have to test them, whether or not they are truly clinical relevant. Otherwise, it could be interest of your particular model. It's nothing you would be interested at all. So then we're using our own TMA at WPC, or using Misha Beltran's and others clinical cohort. We prove that indeed HP1 alpha is significantly higher in clinical NEPC. So of course we went on to did a number of functional study. So this is not a major thing. So you can see this is a kind of using a particular NEPC cell line. You do all sort of cell line assay. And the good news is seems knocking down affect a major effect like cell viability, cell numbers, colony formation, DNA synthesis, so on and so forth. And also knocking down and induce NEPC cell death and apoptosis. More interestingly, then actually you would see the in vivo effect as well. So that gives you a little bit more confidence. But knocking down, in many cases, do give you a positive result. And then the question is, if you overexpression the gene in the adenocarcinoma cell line, what do you see? And in this case, she does see the AE marker goes up after gene expression. So that makes us really interested in this gene a little bit more. And initially, the rationale for us to pick up this gene, one of the rationale is kind of they interact or likely interact with trimethylated H3K9. And then actually in AEPC cell line, the two actually show a really good positive correlation. So meanwhile, we also found overexpression HP1 alpha can inhibit androgen receptor and REST. And it was kind of REST and AR are typical transcription factor for adenocarcinoma. In order to do the transdifferentiation, this typical transcription factor has to be knocking down. And then, and we also find increased H3K9 modification on master lineage specific transcription factors. Actually, AR and the REST promoter region was highly occupied. So moreover, when we're knocking down HP1 alpha, we inhibit the mitosis and DNA damage repairing mechanism. To cut the long story short, actually we are convinced since HP1 alpha is certainly can affect through kind of suppression AR and the REST, and probably play a major role for the transdifferentiation. Actually, first of all, it's a dedifferentiation. So of course, HP1 alpha also affect mitosis, DNA repairing. That's obviously would contribute to NEPC progression. So it's not only just about aggressiveness, actually about initiation and transdifferentiation. So if what we see is true, so this could be the early kind of signal. Whether or not they can be detected in the blood as a biomarker or something, or histone chromatin signature could be something we're looking for. We don't know yet, but the xenograph model probably gave you a good place to begin with. So if we could inhibit HP1 alpha, the idea is you probably not only just affect NEPC, probably you could prevent the transdifferentiation. So in the lab, the other efforts is kind of for drug development. So this is a collaboration with Art Chekhov and Michael Shen and Nada. So then That's actually, this is not something I'm good at. It's collaborators, they are marvelous. They get this set up, the pipeline. They use in silicon modeling system, and for the lab screening, in vitro and in vivo, that's mainly happen in our lab. So we have a crystal structure, and we did initial experimental test, mainly focus on cell-based screening, and to see whether or not they could have any biological consequence. The second one is trying to see any small molecule can specifically binding to the target. So again, I don't want to spend too much time. We move pretty fast actually. So we already have a number of customer-made after all the known small molecule inhibitor and screening for multiple round. Then we have a customer-made ones, and this is 6 of them actually show reasonable IC50. And then more interestingly, when you using non-AEPC cell line adenocarcinoma, they don't show any toxicity. Actually, another news is you're knocking down HB1 alpha in mice, and that doesn't give you significant phenotype. That probably suggests that this gene knocking down may not have a significant side effect. This is the kind of binding assay. And then in any case, so we are still working on this one. We got a good summary. We still modify small molecule. So this is the kind of initially this is the story I want to mainly share. Since I have more time, I'm speaking little faster and move to the second story, which probably excite you a little bit more. So anyway, the second story we said it was targeting some fundamental cancer characteristics. In this case, we talk about MCT4. So in cancer field, we all feel we're doing pretty good, but when we're thinking a little bit deeper, we know we are not that good at all. So we're still not winning the war. So it's hard to find, somehow the good excuse. But one of the things we feel pretty okay to say, why we are not doing so well. Normally we will say because cancer, not because we're not so smart. We are very smart, but cancer is too complicated. And with too many genes, too many pathways, and so on and so forth. So quite a number of years ago, and when Stephen Chow joined me, so this is a marvelous student, and because he's so smart, I start to challenging him. I said, you cannot just pick up a gene and do a project, wrap up a few papers, then you go and leave all the, something behind. And then I said, you got to find a good reason to target on something worth your efforts. And then as we know, instead of looking for some unique features here and there, we say, go back to look at Hallmark of Cancer, because that has been considered as a Bible of cancer field. And then that time is around 2012. And then we look at this one, so we know, and Robert Weinberger and Douglas Anahan and came up with 6 hallmarker, and that was 20 years ago. 10 years later, they became smarter, that became 10. So our question to this guy is that if they are still strong enough to came up with another version of Hallmark of Cancer in 2020, and how many hallmark they have to put on. And then all the students that time, I think it was everybody say, hey, more than 15, more than 20, and the answer is the number of hallmark going to increase because we became more knowledgeable. And so this is kind of really troublesome to me, because if you consider hallmark of cancer is a fundamental of the cancer, so the number of the fundamental shouldn't be increased, because one of the philosopher back to 2,500 years ago, this Dahl, basically said the biggest rule should be the simplest one. And we, as a scientist, we are good at complicated the scenario. Similarly, for cancer, and we know these things, the cancer, it used to be just lung cancer, prostate cancer, ovarian cancer. Now we became so smart, we know the morphological subtype. Now we even smarter, we know the molecular subtype. Then the cancer type became thousands different type of real cancer. The question is, are we in general heading up or heading down? Maybe both direction is right, but I'm not so sure. So then we push him back to say, okay, look at the hallmark of cancer. Can you, have a different look, and trying to find answers for this question? Which of these are truly unique and fundamental to cancer? So the hallmark of cancer described by the guru in the field, and mainly most aspect is to describe the phenotype of cancer, describe the aggressiveness of cancer, and it was kind of, and also they put them in the ring. So are they equally important? Are they unique to cancer? All those things was kind of hardly to answer. I did not have an answer, as well. So Stephen Chow was so smart, and he came up with the first hypothesis, and based on the description we had many years ago from the observation of your clinical fellow, the cancer pretty often living in the lymph node, just like a criminal living in the police station. So then, how, and the criminal could make friends with a policeman. So we said they have to wear their uniform. They have to speak in the language. We describe that phenomena as epithelial immune cell transition. Now you even mess up EMT and neuroendocrine transdifferentiation, epithelial mesenchyme transition. We came up with an epithelial immune cell transition, and then everybody hate us anyway. So then we thought, and if that hypothesis was true, and it was kind of what could be the significance of that hypothesis. And we know in our body, many of potential cancer cell could happen, but only those know how to communicate with the immune cells, they could survive. So if they don't have EIT property, they get killed. So we never really noticed that. Only those cancer cell has EIT property, they know how to talk back, and doing right reciprocal interaction, they can survive. So if so this is relevant to survival and death issue. Is that important? We're also trying to say, like a cancer stem cell, the concept is good, but the marker is random. And then if EIT is necessary for cancer cell to survive, EIT could be a true marker for cancer stem cell. Of course, this concept affects immune therapy and so on and so forth. So we feel pretty good. But on the other hand, we feel bad. When we look at how many immune genes could be turned on by cancer cells, a ton of them. And too many of them. Now the question is, what is the major communication code cancer use to talking to immune cells? Again, we did not have an answer. But could be the practical things, right? And PD-1, PD-L1, that's a minority as well. It was kind of many cancer cells even don't use that mechanism. And also not only T cell, you have B cell, you have macrophage, you have dendritic cells, so on and so forth. So then the student was so smart, right? That's really cool. And then he observed the other two phenomena that's universally associated with basically all solid cancer. So one is kind of, it seems all solid cancer must living in immune deficiency microenvironment. And at one time, we're trying to say cancer actually is a localized immune deficiency disease. Again, many people hate us. So, but anyway, we did not say too much. And then the second phenomenon we found, it seems in this tumor microenvironment, actually they often are acidic. Acidic microenvironment actually generated by cancer. So cancer is an inner player. They are responsible for this acidic microenvironment. Of course, the acidic microenvironment was kind of normally is ignored for years. But back then, Stephen asked the question, is there any connection between the lower pH and the immune suppressive tumor microenvironment? And then from biology point of view, before any evidence, I was convinced. I was thinking, wow, that's really cool, because you lower the pH, and what happened to the normal healthy cells? Because that simply can induce de-differentiation of all sorts of cells. The de-differentiation actually can change in the landscape of gene expression, can simply means loss of cell function. Like we know that if you basically lose the differentiation, how you can function normally. So Stephen actually came up with the idea to say, okay, now cancer-generated lactic acid is not a garbage anymore. Probably has a main meaning, and that's mainly for suppress anti-cancer immunity. Of course, through this process, actually it is a one stone kill two birds. Through this, no matter it's through glycolysis pathway or glutamate nolysis pathway, actually no matter you eat veggies or meat, and they all have a pathway. Amino acid can give you lactic acid. Glucose can give you lactic acid, so it doesn't matter you eat meat or veggie. So that they all can go that pathway. So in this case, we say if you cancer cell and generate lactic acid, and then that can, one way to generate the survive environment that prevent from immune cell killing. But meanwhile, they generated a lot of building block, allow them to make offspring. So you make offspring, you become bigger, and then you got to make a room for them. So through this specific, kind of metabolic pathway, actually, the mainly is not aim for generated ATP. It's mainly generated offspring and also make a room for them to live. So this is the kind of things happened multiple years ago. So if this hypothesis was right, then the idea is, can you really blocking this lactic acid transporting, not really targeting glycolysis only or targeting glutamate nolysis only, because they will shift. Cancer cells are so smart, right? They have a different machinery. They can shift around. So the idea is, if you blocking the backbone, what would happen? Cancer cells only have two choices. One, they're so smart. They realize I cannot generate too much lactic acid, and if so, I couldn't get rid of them. Then that will induce intracellular pH low. That will cause suicide, which is not bad for us. They kill themselves. We save a lot of energy there. And or they figure out they cannot generate lactic acid anymore. Then this is kind of an inner factor. Then our system will buffer in and changing the pathological environment into a physiology condition, then all those cells, including immune cell, can return to the normal function. So that's actually the idea. The main idea is to restore anti-cancer immunity, but not directly kill them, but indirectly. So, and then using the prostate cancer model, we realized that both AR positive CRPC and NEPC, and actually through this progression, the glycolysis pathway are significantly high. Although in the hormonal naive state, the glycolysis is not necessarily always there. So then you probably from a clinical practice, you also know some of them actually is not PET kind of sensitive. But regardless of what happened, once they became a more aggressive stage, and then cancer, and then their glycolysis activity is really high. Then Stephen identified actually in prostate cancer, actually the transporter is a MCT4. The MCT4 actually was kind of major one, not only in prostate cancer, in many other advanced cancer, and MCT4 also is true. So the defined hypothesis is, can we blocking the MCT4 transporting? If that's the case, then that's more specific, right? That can be practically done. So, and he actually had antisense, and basically when he shot down the, using antisense number 1, number 14, which are the two patent one, so then he can see significant, inhibition of lactic acid secretion, intracellular lactate actually increase, glucose consumption became undetectable. If you look at the entire pathway, that totally got shut down. So we see the proliferation, we see the tumor volume, but the real question we want to ask, and can you, kind of restore anti-cancer immunity, after knocking down MCT4? So he actually using two models to do this. This is hard. We all know, particularly use a xenograph and to study cancer cell and immune cell interaction almost impossible. But we make some impossible possible here, and then we use two examples. So one example actually was very simple. We put PC3 cell in nude mice. The nude mice, we know that you don't have T cell, but you have other cells like NK cell. So then because the cell line is not like a human cancer anymore, so they can living in partial immune deficiency mice. So then we actually grow the PC3 in nude mice. We detect, after knocking down MCT4, and then you can see, the NK cell actually, the number actually increased. So that was not a good model. We don't like a PC3 cell. We don't like a nude mice that well. And the second model, that was really interesting. The second model, actually, we use a first generation patient-derived xenograft model. So PDX models, and again, because I'm working on a PDX model for 20 years, so every time you find your friends will challenge you, say, hey, YZ, how are you going to use your PDX to study cancer immunity? You know that I cannot in most cases, but any statement is conditional, okay? So when we're talking about conventionally, we're talking about PDX model, we're talking about the patient tumor grew in the mice, transplanted for multiple generation. So the human stroma component totally get lost. So then you only have human cancer mixed with, the mouse stroma. Then if the mouse is an immunodeficiency one, you don't have enough immune background. So that's obviously is a drawback for us to use such model to study cancer immune interaction, unless you bring human immune cell back. And even for that effort, we call humanized mice. So it's very hard to get a perfect model. You never really get it because you never really can fully recover human immune system in the mice. That will kill the mice, obviously. So you can only recover them partially, for sure. But what we notice, because we do a whole lot of PDX, we notice that when you graft a tumor chunk into not a subcutaneous site, the subcutaneous site many cells cannot survive, then that may not be so good. But what we do, we graft a tumor chunk and the renal capsule of skin mice. And what happened to those grafts actually was kind of not only cancer cell can survive and may forever, and actually human stroma cell and particular those immune cells can last from weeks to 2 months almost. So that gives you a really good window, and within a few weeks after your grafting, the human immune cell that originally associated with cancer actually survive and functionally there. And then they actually, that was kind of, they interact with their de novo cancer. So what we found, actually in the control, you can see control graft, first generation, you can see human, all human Ki67, human CD8, human CD40, 45. You can see human cells actually is there. And what if we treat with the MCT4 inhibitor, in this case the antisense, you can find in the treated group, actually the cell number increase, particularly you see the overall lymphocyte, particular the CD8 number increased. So this model is hard to study, but it's so real because this is not an artificial one. This is a human original one. So I still have 1 minute to finish this one, and so I got a really marvelous young student 2 years ago. Francisco probably did not know her. She was 19 years old. Then this is a student I trying to play game with when they were young. And Stephen, when Stephen working with me for all the hypothesis, that time he was young. Now I don't play with him anymore. So Judy, and there was a summer student came to the lab. I assigned her a task other than the experimental task. I said, you know what? In our life is not easy. Although we have an immune system intact here, but we have to deal with bacteria, virus, parasites, sometimes cancer, and sometimes even the pregnant mother have to dealing with a fetus. And the fetus is a foreigner too, thinking about 50% it's not belong to the mother. Okay, they all foreigners. So the question I asked her, I said, go back to check the literature. What if then is there any kind of common immune anti kind of immune invasion kind of a mechanism they can use? If so, and that's from an evolution conservation point of view, that could be really important. Instead of looking for very, very unique, very, very special things like all the experts would do, we're trying to say what is the common things. If so, is that going to be more interesting? So she actually came up with a paper published early this year. I'm so proud of her. So she found only 2 things that the pathogen and the cancer shared. It's kind of lactic acid generation. The second one is a dormancy. And the dormancy is kind of the mother's nature. It's not acquired ability. And not in normal world, when the environment is not so good, they actually can go to sleep and go to the level, the minimum level to bother the host. They pretty often they succeed, and they once the temperature come back, everything goes well, they recurring again. Cancer exactly the same thing. We are so successful in terms of knocking down the proliferative cancer cell, but we are so incapable dealing with the dormant thing. The dormant cancer cell truly give you the recurrence and the metastasis. Actually, this is the common two of the major things, and she identified. I'm so proud with that, and then it seems we learn new lesson, but it actually was all kind of said by old wisdom. So this is the first one. It's a kind of a Dao philosopher, and basically he said 大道至简 means the most significant rules should be the simplest one. And then I don't have to tell you, and Newton, Einstein basically say more or less the same thing. So then you can see in the overall, we start from modeling the disease, and I believe the good model give you truly a good platform for discovery. And I feel concerned to use a human tissue, human sample for discovery directly because that's too complicated. Perhaps need a larger patient sample. But if you have a simplified version, you have some discovery, then allow you to go back. Once you discover, you know what you're looking for, then you can go to the complexity to find your finding as true, as relevant or not. And then, basically along the line, you push them forward. So I have to thank, and Francisco actually is, I'm so proud of him. And he stayed in my lab for a few years, and he actually was the most productive and as a really nice person I ever have. And it was kind of for this project I'm talking about Stephen Chow, Xinpei, and Dong, and Hui is mainly for the PDX. I have a number of the good collaborators, and Judy is not here. And quite a number of them you see, and a lot of people with young faces. So and I like those young people, and they never really can get contamination. They are so brave to come up with new ideas. I give them a chance to dance. That's I stop here, and it's 48 minutes, 3 minutes past.
So great talk, thank you so much. Elena, please.
Okay, great talk. I have a comment and a question. The comment is about your PDX humanized model. Mm-hmm. Okay, you are able to maintain the immune cells that were inside the tumor at the moment of the transplant, but we know that the tumor continuously to recruit novel immune cells from the bone marrow. And so this is a criticism from your model, because you cannot maintain this tumor bone marrow crosstalk. Yeah. And this is the comment. And then the question was, you show in both your models, the PDX one and the PC3 in the nude mice, that you recruit, in one case the NK cells, and in the second case you increase the T cell number. But I didn't find the data on the tumor growth. It was reduced after the inhibition of MCT4, or?
Yes, so I did not show the data. So okay. that was kind of the tumor volume and the growth actually reduced, but not that dramatic. Okay. It's mathematically significant. We found, I did not show that because the data was not there. And then the reason is I think that was knocking down many occasions you can affect a certain tumor growth. And to me, that's not so dramatic. And I think the MCT4 strategy would do better if you give it a longer period, because this is not like a chemo drug or something. You get acute killing. This is something you're dealing with a chronic disease. you got a chronic strategy and gradually, take care of the cancer. But as a matter of fact, and the tumor volume get affected.
And have you ever thought about doing something in syngeneic immune competent mouse models?
Of course.
Just to see.
Of course. So then actually we just in Edinburgh, I talking to a guy who trained by Charles Sawyer. So they have a high make, mouse cell line. Actually, I had the cell line already. I just want to chat with him what the tumor look like. I'm not totally convinced that that's a good model, but for cancer and immune interaction, I'm okay with that. I just, definitely is the case. The reason we did not show here, because most of the in vivo work we done using MCT4 antisense. This antisense is a human specific, doesn't work for mice. So then, but the work we are doing for MCT4 small molecule inhibitor, actually we just file a patent now. So that actually could cross the different species.
And the very last question,
Yeah,
do you
go
know
ahead.
it is selectively expressed by the tumor, or maybe MCT4 can be expressed also by some stromal cells or immune cells, and so they can be off targets,
Of course.
positive or
Of
negative
course,
off targets?
of course, of course. So actually it was kind of cancer somehow is really interesting. The lymphocyte mainly are using MCT1. Okay, so it's not the same. So, that like AstraZeneca has a program targeting MCT1, and it was kind of they start to focus on leukemia lymphoma type of disease. Actually, very troublesome when it comes to the solid tumor, because imagine that you're targeting a solid tumor. First of all, you may inhibit normal lymphocyte proliferation.
Yes.
So anyway, but for lymphoma, perfect, fine.
Okay.
And then that's all right. MCT4 is a good story, because MCT4, although you can detect them in other cell type, it seems in cancer, it seems that's the most common one.
Okay.
And expressed. We couldn't find any other players has an equal appearance or importance. Yeah.
Thank you.
Yeah, I like your comment, and we talk about that later on, because it was kind of systemically how to provide new immune cells. But for short term of study, the PDX, the first generation, actually, I love it. I'll tell you why later on.
Thank
okay.
Yeah.
So it's funny you mentioned NCT1, because just weeks ago, last Monday, I heard a talk from Sean Morrison who was talking about MCT1, but that is melanoma, and was mentioning the same AstraZeneca inhibitor you were talking about.
Oh.
But my understanding of the two talks is that one of the functions that, along with the uptake of lactate, they also increase proton pump, the proton inside the cell. So that's why the pH change.
Yeah.
So this is perhaps the commonality between the MCT function in this particular setting. So and that is so is the acidic environment that perhaps is as you start your beginning. So is the what they make the environment so immune suppressive.
Yeah, so this is the kind of, I often, because maybe I'm totally wrong, I often trying to thinking about something, pretend cancer also thinking that way. Just trying to say, if cancer, like this smart criminal, always smarter, somehow. First of all, they have to know, like all the kind of life, the first thing is how to survive, and then talking about aggressiveness later on. So I myself is an immigrant to North America, I clearly know the survival mechanism is the most important one. Otherwise, you don't have enough to, right?
Progress.
Yeah, no way. So then, so I intended to thinking about, the lactic acid generation, probably in this, not in the dormant phase though, it's in the proliferative cases. This is probably more generic, more powerful things, simply changing the physiology condition in the pathological condition. In the pathological condition, who really can function normally? No matter you are a professor, you are a policeman, you are a doctor, everybody have to got something trouble to deal with. So that's actually called dysfunctional process, de-differentiation. So I think that's probably kind of a more global kind of things. And also I believe this is probably the upstream of hypoxia, for example, because people ask me who is upstream, who is downstream. And I said, I regard you have oxygen or not, and you often have lactic acid generation. But on the other hand, and you have acidic microenvironment, actually you reduce oxygen concentration. So you tell me who is more important. Some people say this is chicken, egg chicken, but to me it's not. It was upstream. So lactic acid probably is the main thing. Of course there are other mechanisms. Maybe PD-L1 also kick in, maybe B7, H4, H3 also kick in different location. Just like we have many soldiers, there's just a different period and you assign different soldier to work with your army. Yeah.
So and also think that the differentiation can also increase your potential because you are more plastic, so you can become more adapt to the environment, so you can respond to the different environment. You have more chance to respond to the environment.
But for depend on
even an adverse one.
Yeah, but I would think cancer cell. People may think cancer cell may get some advantage by living in the acidic microenvironment. I have a slightly different view on that. I think cancer cell probably also suffer in the acidic microenvironment, but the suffer has a price. The price is they have a life. So you're thinking about we culture cells in the petri dish. So why we don't culture them in the acidic pH? Because they don't really grow that well, right? So so we're trying to give them a pH, physiology pH, so we make them happier. So cancer cell probably also suffer from the pathological condition, but we all suffer. We suffer, and sometimes we work hard, we suffer, and we have a meaning for that, or why we suffer. But cancer cells suffer mainly for the life, for survival. And the other consequence, whether or not they have, other advantage, some of them might, because the de-differentiated, cells may have, the enhanced, the angiogenesis, or perhaps give you more plasticity to move around. So like invasion and metastasis, that might be. But overall, I think cancer cell has to. They don't have a better choice.
Another question?
A lot of people talking about de-differentiation, redifferentiation. That's emphasizing a little bit because in our model we clearly see that. In the adenocarcinoma model, and if you do androgen ablation, you will see the differentiation marker first of all get lost. De-differentiation is a very major mechanism for survival. And then that's actually, you could call them EMT, the neuroendocrine transdifferentiation. Some may not, but they just from a more differentiated one, undergoing less differentiation one. I don't know whether or not you find a marker to say this EMT or stemness or something. Any depend on which corner you look at, you will see that. But the bigger picture is a differentiation and redifferentiation.
Just a quick question. In Francesco's talk, he talked about the presence of enzalutamide interaction and the neuroendocrine sort of stimulation almost. In your PDX models of neuroendocrine, can you accelerate the process by drug intervention?
Yeah. So actually, none of those drugs are as good as a castration in mice, because the mice give you perfect androgen ablation. It's kind of that in mouse. Almost 100% of them come from testes, not like human. Human, you have 10% from adrenal gland. So this is the mouse system. If you castrate the mice, that's the maximum androgen ablation. So none of the MDV or AB would give you same profound. The androgen ablation is the most effective one. This is perfect for the modeling. Yeah.
Okay, thank you so much.
Thank you. Great.
Okay, so I'm going to talk about my postdoc project, which is on the long non-coding RNA near-1 and the long non-coding RNA landscape in NEPC. So I don't need to explain this to you, but just to demonstrate our hypothesis was, are long non-coding RNAs involved in the initiation and progression of this disease? So long non-coding RNAs are a huge part of our genome. We have almost 60,000 of them in our DNA. Less than 5% of these have been functionally characterized, and they are hugely functionally diverse. So you can see some examples here. Depending on their cellular localization and their cell type, they have different roles. So some, for example, in the cytoplasm can act as microRNA precursors, and some in the nucleus can be epigenetic modulators. Importantly, these regions of our DNA evolve rapidly, and as such, evolutionary conservation is extremely poor. Less than 100 of these, nearly 60,000, are conserved throughout evolution, and these are thought to be associated with more fundamental biological processes. And importantly, many of these are expressed in a cell type and disease-specific manner, making them really attractive as potential drug targets. So there have been several studies of long non-coding RNAs in NEPC. This is just a few examples. But they have different approaches to us, and actually none of them have done really comprehensive studies, starting from big data sets that are also functional. So some also we haven't been able to corroborate their data in other data sets, and there are a couple of examples here. So our approach is slightly different. And in order to address our hypothesis, we used the patient-derived xenograft that you just heard about from Wesley's lab. And this model is 331 to 331R, which is a hormone-sensitive prostatic adenocarcinoma. When the testosterone implant is removed, this mouse model transdifferentiates into NEPC. And we sent some of these samples off for RNA sequencing, and we got a huge list of non-coding RNAs which were upregulated in the NEPC model. We ranked this based on various criteria, which narrowed us down from just over 15,000 to just 26 transcripts. And these were ranked based also on evolutionary conservation using CLAR analysis, which is a bioinformatic tool which was developed by our collaborator at the Weizmann Institute. And based on these 17 species that had orthologs, we had these RNAs which we called NEARs, so neuroendocrine-associated RNAs 1 through 26. And we screened our RNAs for expression in cell lines. So you can see here on the left-hand side, this is a screen of various NEARs that we found in our RNA sequencing dataset. Importantly, our most conserved transcript, NEAR1, is also the most highly expressed in the NEPC xenograft model. So we moved forward with that. Also, bioinformatically, I looked at patient data from the TAMRIC dataset of prostate adenocarcinoma patients. And even though this is a relatively small number of patients, you can see even at the earlier stages of disease, high expression of NEAR1 correlates with a poorer overall survival. To ensure that this transcript was clinically relevant, we also used data from microarrays. So here on the left, this is further patient-derived xenograft models from Weiz's lab. And you can see here a significant increase in expression, although there's a broad range, in NEPC compared to other adenocarcinomas. This is also true in patient biopsy data shown here in the middle. Between CRPC and NEPC, we can differentiate there. There's a higher level of expression in NEPC. And in our hands, these are samples provided by Holly's group in Surrey. RNA was isolated by a collaborator in Switzerland, and the PCR that we did here showed a high level of expression even in these archived tissues. So then we looked at a panel of prostate cancers and one non-neoplastic cell line, which is RWPE1. And you can see here in our NEPC-like model, PC3, a high level of expression of NEO1, and this was conserved in murine cell lines kindly provided by Mario and Elena, where we see the same expression pattern. One of the other things we do is to check cellular localization of these RNAs. This is essentially to show us whether we can target them with siRNAs, but also gives us a rough indication of where we could find a mechanism of action. On the left-hand side, this is the human data. The PC3 cells show an 85% cytoplasmic localization. This is different in the murine cell line, and we see around a 50-50 level of expression. So that may indicate that there's a different functional mechanism of action there. So moving forward just with the PC3 cell line, the human cell line, we did some siRNA knockdowns with 3 different sequences. You can see by qPCR we get a reasonable level of knockdown, which then translates to a reduction in the number of metabolically active cells shown by NTT assay here on the right-hand side. We then looked to see whether this reduction in the number of cells was due to cell death. So I did some caspase activation assays. I did this in PC3 and also RWPE1 cells. And you can see here with NEO1 knockdown, you significantly increase the level of caspase B7 activation. And just to show you that these cells are pretty unhappy through bright field microscopy. One of the next things we did was to determine whether NEO1 also had a role in metastases and invasion. So we did this with siRNAs at an earlier time point at a lower dose so that I wasn't skewing my data. You can see that I got a significant knockdown by qPCR here on the left, but I wasn't skewing my data unfairly with reduction of metabolically active cells shown here on the right. Using Boyden chambers through Matrigel, this is done in collaboration with Wesley's group, you can see that there is a significant reduction in the amount of cells that are able to migrate through that Matrigel. So NEO1 also has a role in invasion. And using patient data, you can see here that NEO1 expression is higher in liver metastases, which are associated with the worst prognosis. And this may be an advantage for antisense oligonucleotide development because antisense oligos tend to accumulate in the liver. So that's all well and good, but how does NEO1 work? So we looked at the literature, and there was a study in 2015 in pancreatic differentiation that showed that NEO1 could recruit SMAD2/3 to the promoter of FOXO2, which results in its transcription. And there was another study 2 years later demonstrating that FOXO2 levels of expression were higher in the PC3 and H660 NEPC cell lines. And FOXO2 has also been shown to be a molecular marker of NEPC, but there's been no functional studies on that to date. Bioinformatically, we found that FOXO2 strongly correlates with NEO1 expression. And when I knock down NEO1 using siRNAs, I also see a reduction in FOXO2 at the mRNA level. So to determine whether the mechanism of action was the same, our collaborator, who was actually the group that published the paper in pancreatic cells, they did some RNA immunoprecipitation assays and showed the same binding of NEO1 to SMAD2/3. This was interesting because the localization was different in their study, and they found that NEO1 was localized to the nucleus, and indeed that's where FOXO2 was found. But this was also seen to be true in the PC3 cell line. When you knock down NEO1 with siRNA, we also saw a reduction of FOXO2 at the protein level, which was conserved in the murine cell lines as well. We then validated whether this was a likely mechanism using data from the PDX model, and you can see that the FOXO2 transcriptional program here is active in the NEPC model, but not in the adenocarcinoma model. And over time, NEO1 and FOXO2 both emerge at 12 weeks post-castration and are highly present in the NEPC model. We then did some stable knockdowns of NEO1 in PC3 cells using lentiviral shRNAs, and this was to determine whether or not this was true in vivo. So on the left-hand side, you can see that we achieved a stable knockdown of NER1, and we saw a small reduction, which was still significant in FOXO2, in the cell line as well. These were then implanted into nude mice, again in Canada, and you can see here that the same sort of proliferative phenotype is observed, so there was a delay in tumour formation and a reduction in tumour volume. So we still don't really know how NER1 reduces proliferation, and we've done some more RNA sequencing on the lentiviral knockdowns and the siRNA cells, and using reactome analysis we found a phenotype associated with interferon signaling, and this is particularly active when you knock down NER1, as you can see on the right hand side. This is not just interferon signaling but also immune response proteins, and we're working on that at the moment. So there was a study in Cell Reports in 2014 which demonstrated in PC3 cells that the interferon gamma receptor was affected. It had a domain missing, and this activated this signaling pathway. And we've looked in the PDX models, and you can see here that there's this sort of pattern of expression where in the NEPC phenotype the upstream proteins in this pathway are almost absent. So we're working on that as our work at the moment, and on a sort of side project I've been working on NER1 as a potential for liquid biopsy. So I've been developing a method in collaboration with the ICR, isolating RNAs from the urine of patients with adenocarcinoma and NEPC. And you can see here this is a very small subset of patients, but we've achieved a signal that's good enough to be able to differentiate in this small cohort between adenocarcinoma and NEPC for NER1, FOXO2 and 2 neuroendocrine markers. And another aspect of this project is whether or not we can target NER1 in vivo. So for many reasons you can't inject siRNAs into in vivo because they need lipid reagents which are toxic, and also siRNAs are not stable enough. They get degraded rapidly in the blood. So I've been designing sequences against NER1. They have these locked nucleic acid regions which specifically bind to the transcript, and they flank an RNase H activating region. There are several of these locked nucleic acids in trials, and at the moment there's a phase 1 clinical trial with a plasma concentration achieved of 97,300 nanograms per mil. When I've been screening these, I've screened about 20 so far. We got 2 that achieve a decent knockdown, so you can see the qPCR here on the left. And on the right hand side, a cell count demonstrating that this phenotype of a reduction in proliferation is observed with these antisense oligos. When I've done some dose responses, you can see here that the IC50 is around 70 nanomolar, and when we compare that to the clinical trial, which is published, theirs translates to about 18 micromolar as an achievable Cmax, so we're well within the clinically achievable range. We have a poster on this as well with a bit more information if you're interested. So in conclusion, we found that NER1 is functional. It has roles in proliferation, invasion and survival. It has potential to be targeted in vivo, that our doses of LNAs are clinically achievable, and that liver accumulation of locked nucleic acids might be of use because NER1 is also found in liver mets. Non-invasive assays show that NER1 could be a potential biomarker, and we're currently working on NER1's mechanism of action, NER1 in urine, and moving the LNAs into NER1.
Very nice presentation. Thank you. Is there a question? So, I'm very intrigued of the interferon gamma regulation. So when you down-regulate the long non-coding RNA, you have an increase in interferon gamma. Does it mean that in presence of your microRNA, do we have an IL-10 production, for example?
Possibly, so it doesn't directly affect interferon gamma levels, but it activates the pathway downstream. So we're not quite sure on the mechanism of how it does that at the moment, but there's also various cytokines that are upregulated in the absence of the long non-coding RNA. So perhaps that is how this is acting.
because also interferon gamma has the interferon family, so have two activities. So one is positive in terms of immune response, but also could be negative because it can induce IDO1, PD-L1. So it has two activities. So it depends on the setting. So but it's very nice observation. It's very nice.
So that could be in terms of therapeutic that may generate some concerns, right? When you instead using systematic method and knocking down, interferon gamma, you probably inhibit innate immunity. Then that actually would be a good thing to have. You don't want to lose that. So I have a small question for you. And
can I just add, but NER1 is expressed only in the neuroendocrine prostate cancer cells. It's not expressed almost anywhere else. It's only expressed like in developing cells of the intestine, right, mainly. But then, if you knock down NER1, you're almost sure to target only the neuroendocrine cancer cells.
So, okay. There is no other question. Thank you so much. Very good.
Okay, thank you very much to the speakers and to Sushila and Mario for chairing the sessions so brilliantly. I hope it's been useful for everyone. And I think in a few minutes, probably, we will have some more refreshments, tea and coffee, possibly to share ideas. And if you have time, we have some surveys that we use to measure how useful these type of events are. So if you could fill them and then leave them at the entrance, I think